Welcome to Yakeen Neet English batch. Good afternoon, everyone. Lecture started, not yet Choco. Good afternoon. Kirti, Bhumika, Tiso, Bhuvneshwari, Akshara, Vanshika, Chaitanya. Good afternoon, everyone. How are you all doing? Shubha Madhyana, ellarigu. Is the audio and video clear? Let me know in the comments. Hello Meghna. Meghna sending me so many grasses. I am doing good. Nalla irukre. Chanagidini. Bagunnanu. Alright. So in the last class we discussed about the tissue systems and we spoke about anatomy of dicot and monocot root. Do you have any doubts in that? Maliha has finished everything. Everything's up to date. Brilliant. Oh, grass is favorite for Ganapati. Do I look like an elephant to you? Do I look like Ganapati? Alright, let's get started. No doubts then. We'll move on. We have a lot to finish today. We have to solve MCQs and all of that. Right? Everybody is ready. Give me a thumbs up and we'll get started real quick. Cool. Let's get started. I'll disable the comments for a little while. So in yesterday's class, we finished anatomy of root. Today we're going to discuss about the anatomy of stem and leaves in both dicots as well as in monocots. Alright, let's get started with dicot stem. What you're looking at here is a transverse, excuse me, a transverse section of a dicot stem. If this is a dicot stem, we take an extremely thin slice of the dicot stem, look at it under the microscope and observe. So when you cut, let's say this stem that we are talking about is cylindrical. So when you cut something that is cylindrical, obviously the section's outline will be circular, right? So we get the circular outline. What we are going to learn right now is one section of it. The TS of dicot stem will look like this, but we will learn only one portion of it, like this much. Whatever we are learning about here, the same thing will apply throughout this section. Okay? So this is basically this, and we will start learning from here. Alright? Now, let's get started. Like yesterday, when we spoke about dicot root or monocot root for that matter, we started from the outside. Epidermis, cortex, endodermis, pericycle, all of that, right? So we'll follow the same thing. We'll start from the outside and go towards the inside. You saw that in monocot root and dicot root, there was not much difference. It was mostly the same, except in dicot root, the pith was small. In monocot's root, the pith was conspicuous. In dicot root, the number of vascular bundles was 2 to 4. In monocot root, it was 6, more than 6. Polyarch condition, right? So today we will learn about dicot stem, monocot stem, their differences. There are a lot of differences between dicot stem and monocot stem. They are very unique. So pay attention, alright? We'll start from the epidermis. When we spoke about the epidermal tissue system yesterday, I told you that epidermis consists of 3 different things. One is epidermal cells, obviously. On the outer surface of the epidermal cells, cuticle may be present in the aerial parts of the plant to reduce transpiration. On the stem, multicellular hair may be present. They are called trichomes. And in young stems, you even see stomata. Okay, so if you're taking a section of a young dicot stem, which is kind of like green in color, it'll be very flexible and soft. In that epidermis, you may find stomata also being present. All right. Now, epidermis, as always, is the outermost layer. Since this is the aerial part of the plant, it can be covered with a thin layer of cuticle. Now, what does this cuticle do? This cuticle is a waxy substance. It will reduce the rate of transpiration. I told you it's like Vaseline, right? Reduce the rate of transpiration. Next. In all stems, hair will not be present. In some stems, it may be present, hair and stomata both. If hair are present, we call them trichomes. What do these trichomes help in? Trichomes also reduce the rate of transpiration. Stomata may be present. If stomata are there, what is its function? The function of stomata is to help in exchange of gases. Okay, that is about the epidermis. Now, what was present inside the epidermis in the root? You have the ground tissue, which is cortex. Even over here, within the epidermis, immediately below the epidermis, you have the cortex. Now, this cortex is divided into 3 different zones. Alright. So, previously we saw in the root cortex was there. Innermost layer of the cortex was called endodermis. Over here also you have endodermis, which is the innermost layer. But apart from the endodermis, the main cortex itself is divided into 2 distinct zones. So, within cortex you have 3 distinct zones. The region of the cortex that lies immediately below the epidermis is called hypodermis. Remember what hypo means? Hypo means below. Epi means above, right? So, the outermost layer is the epidermis. Right below the epidermis, you have a region called hypodermis. Hypo means below. This hypodermis is composed of collenchyma. This collenchyma you will study about only in dicot stem. Remember that. Nowhere else you will learn about collenchyma. So you can see epidermis is a single layer parenchymatous barrel shaped cells. Right below that you have hypodermis that is composed of collenchyma. You can see this red cells layer, this entirely is collenchyma. Obviously, there is more than one layer, so it's multiple layers, a few cell layers in thickness. Right below this hypodermis, you have what is called the general cortex. The general cortex is very similar to the cortex of the root. In the cortex of the root, what was present? In the cortex of the root, we had parenchymatous cells with thin walls and intercellular spaces, right? Even over here, the general cortex will have parenchyma, it will have multiple layers, it will have intercellular spaces and it will have thin walls. Okay, so here also parenchyma is very similar, it has thin walls and intercellular spaces. I already told you yesterday why intercellular spaces should be present, because it will ensure aeration for all of the cells. All of the cells will get access to oxygen to undergo cellular respiration. Okay, so therefore intercellular spaces will be present. This green region that you look at here, that you can see here, this is the general cortex. Now the innermost layer of the cortex, is called your endodermis. The innermost layer of the cortex is called your endodermis. It's the same even in the root. In the root, endodermis was special because in the endodermis, you had radial and tangential walls thickened with the deposition of suberin. It formed Casparian strips there. These Casparian strips are impervious to water. They will not allow water to pass through it. Similarly, here you have something special about endodermis. It's not that they have Casparian strips here, they do not have suberin, but the endodermal cell layer in dicot stems store starch. They are rich in starch. Therefore, we call endodermis itself as the starch sheath. Okay? Endodermis is called starch sheath. Understood? What about the cortex should we remember? Cortex has multiple layers. It has multiple zones. Right below the epidermis is your hypodermis. Hypodermis has many layers. It is made up of collenchyma. Do you find collenchyma anywhere else in the dicot root, stem, leaf, monocot stem, leaf, anywhere? You find it only in the dicot stem. Nowhere else you will find collenchyma. Okay, so remember that. And then basically collenchyma, what does it do? It provides mechanical support and some amount of flexibility to young stems. Right below your hypodermis, collenchymatous hypodermis, is your parenchymatous general cortex. Over here you have thin-walled parenchyma, multiple layers with conspicuous intercellular spaces. Now the innermost layer of this cortex is called endodermis. In the endodermis you will have barrel-shaped cells, and in the endodermis there is it's rich in starch grains, so you call it starch sheath. That's about the cortex region of your dicot stems. Now what will we find inside the endodermis? We find the pericycle. In monocot and dicot roots, in yesterday's class, we saw how the pericycle was single layered. It was made up of parenchyma. Over here, the pericycle has many layers. The pericycle is sclerenchymatous, not parenchymatous, and it forms half-moon shaped structures, semilunar patches. Okay, so semilunar means what? Semilunar means half-moon shaped, like semicircle. Like this. And it is made up of sclerenchyma. I showed you initially over here. This is the transverse section of a dicot stem. Over here, you can see how the vascular bundles are neatly arranged in the form of a ring. So these are your vascular bundles. Okay, they are very well arranged in the form of a ring. Now, wherever vascular bundles are present, over there, xylem will be present towards the inside, phloem will be present towards the outside. In between, because it's dicot, cambium will be present. You can see how in certain regions, vascular bundle is there. Over here, there is no vascular bundle. Here, there is no vascular bundle. There are spaces, right? Between vascular bundles, there are spaces. Wherever vascular bundle is not there, over there, sclerenchymatous pericycle will not be present. If a vascular bundle is present there, right on top of the vascular bundle, it forms like a cap-like structure. Okay, you can see here. This is the pericycle. It is made up of sclerenchyma. It forms a cap-like structure only where your vascular bundles are present. It will be present right above the phloem. See, this is the inside, towards the inside. This is towards the outside. In the vascular bundle, here you have xylem, here you have phloem, here you have cambium. Right above the phloem, it will be present. Because it's present like a cap to the vascular bundles, some books even mention that the pericycle forms a bundle cap. Okay, understood? Pericycle is present. It is sclerenchymatous. It has multiple layers of sclerenchyma. It is present right on top of the phloem wherever vascular bundles are there. It kind of forms a cap-like structure. So we call it bundle cap. Okay. Now, let's talk about dicot, vascular bundles. Now over here, we know that the vascular bundles are very well organized and arranged in the form of a ring. I already showed you that. So in dicot stem, you can see that the vascular bundles are arranged very well in the form of a ring. Now if you look at each vascular bundle, within each vascular bundle, is there xylem and phloem both? If they're both there, what do we call them? We call them conjoined. In roots, we saw that xylem and phloem were present in separate radii, we call them radial vascular bundles there. Over here, they're present in the same radius. Each bundle has both xylem and phloem, so we call it conjoined. Now, because between the xylem and phloem there is a strip of cambium present, we say that it is open type. So, the vascular bundles that you find here are conjoined open type of vascular bundles. Talking about the xylem, let's take a look at where the protoxylem faces. Over here, you can see the smaller protoxylem elements facing towards the center and the larger metaxylem elements facing towards the periphery. This is metaxylem, this is protoxylem. If protoxylem is facing inside towards the pith, what do we call it? We call it end-dark. Here, xylem shows end-dark condition. Now, if you look at this dicot stem, you can see how the pith is very clear, right? This entire central region is the pith. The pith is highly conspicuous. It's a very well-defined pith. From the pith, you can see pith is basically made up of parenchyma. Pith is made up of parenchyma. From the pith, it will seem like this parenchyma that is present in the pith, it will seem like it is radiating out between the spaces. It's radiating out into the spaces between vascular bundles. Just like how we draw sun rays, no? It is radiating outward. So, whatever is radiating outward in between the vascular bundles, we call them medullary rays. We call these medullary rays. The pith and medullary rays are both parenchymatous in nature. Yesterday when we spoke about dicot root and monocot root, we spoke about how in between xylem and phloem in the roots, parenchymatous tissue will be present. What did we call them? Does anybody remember? We call them conjunctive tissue. Like that here, you have something called medullary rays, which is parenchyma that is present between the vascular bundles. Alright. Yeah. So, few layers of radially placed parenchyma cells between vascular bundles will be present. We call them medullary rays. Next, pith. Pith is rounded. We saw that it is very conspicuous and it is made up of parenchyma. Pith, as always, will have intercellular spaces. That's about your dicot stem. If anybody has any doubts, let me know. Can someone tell me the difference between dicot stem and dicot root? In dicot root, unicellular root hair will be present. In dicot stem, multicellular trichomes may be present. Talking about the epidermis. In dicot roots, cuticle will be absent. In dicot stem, cuticle may be present. In dicot root, the cortex is not differentiated into different zones. In dicot stem, you see a differentiation into 3 different zones. In dicot root, there is no hypodermis. In dicot stem, you have a hypodermis. In dicot root, pith is not very prominent. In dicot stem, pith is very prominent. In dicot root, endodermis has Casparian strips. Casparian strips are absent in dicot stem. In dicot stem, you have starch-rich layer. We call it starch sheath. That is not there in dicot root. In dicot root, pericycle is single-layered parenchymatous. In dicot stem, Pericycle is multi-layered. It is made up of sclerenchyma. In dicot root, you do not find collenchyma. In dicot stems hypodermis, you find collenchyma. The vascular bundles are radially arranged in dicot root. It is conjoint open type in dicot stem. Xylem is exarch in dicot root. Xylem is endarch in dicot stem. Alright, so these are the differences between your dicot stem and dicot root. Sclerenchyma between vascular bundles. Kirtana, there's no sclerenchyma between vascular bundles, it's parenchyma. Right, Shauritra has another difference, epiblemma. In root, you can call the epidermis as epiblemma, but in stem, we don't call it. Okay. Not necessary, Daniel. I'll tell you whenever you have to make notes, I'll tell you what you need to do. Okay. Now, let's move on and talk about monocot stem. Monocot stem is very easy. No complications there. Okay. Okay, I'll keep those emojis on, but the chat off. We'll talk about the monocot stem now. Monocots include all your grasses, bamboo, all of that. Okay. So, if you take a section of a monocot stem, even then you will get a circular outline of the transverse section, right? This is the circular outline that you will get. If you take one portion of that circular outline, we arrive at this, and this is what we will learn. As always, the outermost layer is what is called the epidermis. Epidermis always is parenchymatous. Even here it is parenchymatous. It is made up of a single layer of epidermis that is barrel shaped. Okay. Over here, the cortex is differentiated. Okay, you have a hypodermis and you have a general cortex. But here you do not find endodermis, you do not find pericycle, you do not find pith. In dicot stem, we saw how the vascular bundles were very neatly arranged in the form of a ring. In monocot stem, the vascular bundles are scattered in the cortex. So, right below the epidermis, you will have hypodermis. So, this hypodermis here is made up of sclerenchyma. And this hypodermis is also multiple layered. Okay. So, here you can see the hypodermis. Hypodermis is a few layers in thickness. It is made up of sclerenchyma. Right after the hypodermis, you have parenchymatous ground tissue. Again, this has many layers. Okay. Now let's talk about vascular bundles, because we have nothing to talk about endodermis, pericycle. They don't exist here because in the general cortex itself, vascular bundles are scattered. Okay. So here, vascular bundles are scattered. You can see that. Look at the size of the vascular bundles. Look at the size of the vascular bundles here. It's not uniform. But if you compare this with the vascular bundles in dicot stem that we studied about, over there, all of the vascular bundles were nearly of the same size. Over here, that is not the case. Notice how the centrally located vascular bundles are much larger in size compared to the peripheral ones. In monocot stems, you see that the vascular bundle size varies depending upon where it is located. If it is in the center, it will look larger. If it is at the periphery, it will look small. You may have seen this in schools and colleges. If you ever made sections of monocot stems, or if you had permanent slides, you may have seen. This is very beautiful to look at under the microscope. These vascular bundles actually look like skulls. Okay, like two eyes and one mouth. It looks like that. So they're very pretty to look at. If I can find a nice beautiful slide of this, I will share it with you on the group today. Okay. Yeah. So the vascular bundles are scattered. Scattered, I haven't written. They are scattered in the cortex. Their size varies. The peripheral ones are smaller and the centrally located ones are much larger. What kind of vascular bundle do we find here? We find conjoint type of vascular bundle where both xylem and phloem is present within the same bundle, and the cambium is absent. The strip of cambium is absent between xylem and phloem. Therefore, we call it conjoint closed type of vascular bundle. The xylem here, the vascular bundles here in some plants will contain cavities which will store water containing cavities. You know that the hypodermis will offer mechanical support, right? Because it's made up of sclerenchyma. Now the same sclerenchyma is present around the vascular bundles here. So here I will show you. These are the xylem elements. It looks like one face. And then here you will have phloem. Surrounding that, you will have sclerenchyma. Since the sclerenchyma is present around the vascular bundles, it's forming a sheath around the vascular bundles, we call it bundle sheath. What did we call sclerenchyma in dicot stem? We called it bundle cap. Where is sclerenchyma present over there? It is present right on top of the phloem like a cap. Over here it's present surrounding the xylem and phloem. It's forming a sheath. So we call it bundle sheath. Alright. This we had already studied. In monocots, phloem parenchyma is absent. Remember when we spoke about phloem, I told you it's an important point. You have to make note of it. So phloem parenchyma, you don't find it in monocots. Remember, phloem has 4 different types of cells in it. You have sieve tube elements, you have companion cells, you have phloem parenchyma, and you have phloem fibers. So phloem parenchyma component of phloem will not be seen in monocots. This is an important point. I want you to remember this always. Okay? Yeah, that's about monocot stem. Very simple. Human skull is beautiful, of course it is. Why is it not, Shahid? Everything about humans, everything about life is beautiful. Cool. Now let's differentiate dicot stem and monocot stem. I'll ask you for questions. You will have to type the answers in the comments. Okay. Pratik, I can share the difference table. It will take me like 10 minutes to do it, but I want you to do it. I want you to do the work. Arun is asking, I will read your notes or NCERT both. Please help me. Read both. Okay, so now the differences between dicots, dicot stem and monocot stem. First difference between dicot stem and monocot stem. We'll start from the epidermis. The epidermis in dicot stem generally has trichomes, but in monocots usually it's glabrous. You do not find the trichomes. Okay. Second, let's come to the hypodermis. What is the hypodermis in dicot stem made up of? It is made up of collenchyma. What is the hypodermis in monocots made up of? It is made up of sclerenchyma. In dicot stem, you have a clear differentiation of endodermis and pericycle. In monocot stem, you do not have a clear differentiation between endodermis and pericycle. In dicot stem, vascular bundles are arranged in the form of a ring. In monocot stem, vascular bundles are scattered in the cortex. In dicot stem, the size of the vascular bundles is nearly the same. In monocot stem, the size of the vascular bundles vary. At the center, you will find larger vascular bundles. Towards the periphery, you will find smaller vascular bundles. In dicot stem, you have conjoined open type of vascular bundle. In monocot stem, you have conjoined closed type of vascular bundle. In dicot stem, you have sclerenchymatous bundle caps present on top of the phloem. In monocot stem, you have sclerenchymatous bundle sheath that covers the vascular bundles entirely. In dicot stem, you have a prominent pith. In monocot stem, you do not have a prominent pith. In dicot stem, you find, what is it called? In monocot stem, you find water cavities in the vascular bundles, water containing cavities. In dicot stem, you do not find that. Collenchyma is present only in dicot stem. Parenchyma is present in both. Sclerenchyma is present in both. But the location is different. Sclerenchyma in monocot stem is present in the hypodermis as well as the bundle sheath. In dicot stem, it is present only in the bundle cap. Okay? Poll questions, Gunjan, we'll do in the end. Oh, one other difference. In monocot stem, you do not find phloem parenchyma. In dicot stem, phloem, you will find parenchyma. In both stems, it is endark. Diya. Is it clear, everyone? Any doubts? We'll have to move on to leaf. Farshida, medullary ray is also only in the dicot stem. Pericycle is not the same in both. There is no pericycle in monocots. Who's that? Ramya. Alright, let's move on. Last part, we'll talk about leaf. First, we'll talk about the dicot leaf. Then we'll talk about monocot leaf. Dicot leaf is also called dorsiventral leaf. Okay, so dicot leaf is also called dorsiventral leaf. When do you call something dorsiventral? When the two surfaces, dorsal and ventral surfaces, don't look alike, they don't appear similar, we call it dorsiventral, right? So if you take, for example, if this is a leaf, okay, this is my son's school magazine. If you think of this as a leaf, okay, you will take a section like this. You will take a section like this. At the center, you will have the midrib. This midrib, within the midrib, you will have vascular bundles. Okay. So, what you're looking at is this. Okay. Look here. Are you getting my point? How did we come to this image of a leaf? If this is the leaf, this is the dorsal surface, this is the ventral surface, this is the upper surface, this is the lower surface. In the center, you will have a midrib. In dicots, you have reticulate venation, so you will have a midrib. This circle here denotes the midrib. So what you're looking at here is this. You're seeing the vascular bundle, the midrib, and one complete portion of one side of the leaf, the other side is half only. Whatever you're learning on this side, the same thing will be present on this side as well. This section is called a vertical section. This kind of section is called a vertical section. What we saw in root and stem, that is transverse section. In transverse plane we cut it. This we cut it longitudinally or vertically. So this is called a vertical section or VS. Okay. Now, Why is dicot leaf dorsiventral? Why is there a difference between the upper and the lower surface? Let's understand. First of all, in dicot leaf, you have epidermis on two sides. In leaves, you will have epidermis on two sides, right? On the upper surface also there is epidermis, on the lower surface also there is epidermis. On the upper surface, the epidermis has a special name. You can call it upper epidermis, but it's technically called adaxial epidermis. Alright, the upper epidermis is called adaxial epidermis. How I remember this is D for direct. So, upper surface you can directly see. So, adaxial epidermis is on top. The lower surface is called the abaxial epidermis. This lower epidermis is called the abaxial epidermis. B for base, it is present towards the base. Upper surface, adaxial epidermis, lower surface, abaxial epidermis. Now the primary difference between the two surfaces of the dicot leaf is in the epidermis. Leaves obviously should have stomata. Through the stomata itself, carbon dioxide will enter the leaves for photosynthesis to take place. But if too many stomata are present, then transpiration rate also will be very high. So what these organisms, in dicot leaves what happens is, as an adaptation, you will find that the number of stomata are more towards the lower surface. On the upper surface or the adaxial epidermis, stomata may be completely absent, or even if it's present, their numbers are negligible compared to the lower epidermis. So in the lower epidermis, you find a large amount of stomata. This is one difference between the dorsal and the ventral side, the adaxial and the abaxial epidermis. So here on the upper epidermis, you find no stomata. On the lower epidermis, you will find stomata. This is the stomatal cavity. And inside this stomata, you will have, this is the stomata. These are the guard cells. This pore is the stomata. So right inside the stomata, there will be a space which is called the substomatal cavity. Sub means below. So substomatal cavity is that space present right below the stomatal aperture or the stomatal apparatus. Alright. So upper epidermis is called adaxial, lower epidermis is called abaxial. Similar to stems, even even leaves are aerial parts of the plant from which water loss can happen by transpiration. So to prevent or reduce the rate of transpiration on both surfaces of the leaf, upper and lower, you can see deposition of cuticle. So cuticle will be present and will be very thick and conspicuous. Compared to the leaf, on the stem it will be slightly thin layer, but in the leaf there will be a very thick layer. Alright, next. Number of stomata. Abaxial epidermis. This lower epidermis will bear more number of stomata compared to the upper epidermis. Adaxial epidermis, upper epidermis may sometimes even lack stomata. Okay, so that is one difference between the upper and lower surface in the epidermis. Cuticle is present in both, there is no difference in that. In both upper and lower, parenchyma is only there, it is barrel shaped. In the upper epidermis, you don't find stomata, or even if you do, it's very small in number. In the lower epidermis, you do find stomata. Alright, now let's talk about the cortex. The cortex in leaves has a special name. We call it mesophyll. Remember, we've studied about sporophyll. When I spoke about sporophyll in plant kingdom, I mentioned that phyll means leaf. Phyllotaxy is about the arrangement of leaves. So phyll means leaves. Meso means what? Meso means in between. So whatever ground tissue is present in between the two epidermal layers, we call that the mesophyll. Okay, so this is called the mesophyll. Now the second difference between the dorsal and ventral sides in a dicot leaf is with respect to the parenchyma in the mesophyll. You know that dicot leaves, or any leaf for that matter, is the photosynthetic organ, right? It is where photosynthesis majorly takes place. So the parenchyma that is present in the mesophyll will have a lot of chloroplasts. They are basically called chlorenchyma because they have abundant chloroplasts for photosynthesis. Now, if you look at the way in which these parenchyma cells containing abundant chloroplasts are arranged, you can see a distinct pattern. Towards the upper epidermis or the adaxial epidermis, the cell's shape is rod shaped. It is columnar, it is rod shaped, and they are arranged very compactly without any intercellular spaces. But if you look at the lower surface, you can see that there are parenchyma cells containing abundant chloroplasts, but their number, shape, arrangement is different. Their shape is almost spherical isodiametric. Their number is less because they are not very compactly arranged, and there are a lot of intercellular spaces present between them. The rod-shaped parenchyma cells are called palisade parenchyma. Palisade is basically referring to its rod shape. Okay, so this is your palisade parenchyma. Over here at the bottom, you have fewer cells that are spherical with a lot of intercellular spaces. That is called spongy parenchyma. Why? Because like a sponge that has a lot of pores and spaces, even here in between the cells, there are a lot of spaces present. Okay, so here you can see there is a difference between the upper and the lower regions of the mesophyll. Towards the adaxial epidermis, you have palisade. Towards the abaxial epidermis, you have spongy parenchyma. You may have noticed how in dicot leaves, many a times the upper surface will appear bright green, very dark green, and the lower surface will have comparatively a lighter shade of green. The reason is because the number of palisade parenchyma cells is more and their arrangement is more compact. That will give it a dark green color. And at the base, near the abaxial epidermis, the number of cells and its spacing is more. That is why it will appear slightly lighter in color. So this is another reason why there is difference between the upper and the lower surface, because though all of these mesophyll cells are involved in photosynthesis, all of them have chloroplasts, how they are arranged, what is their shape, that will vary. Okay, now mesophyll cells definitely will possess chloroplast. I've already told you about epidermis and leaves, right? In leaves, epidermis, chloroplast is not present. It's only present in the guard cells. Apart from guard cells, no other epidermal cells will have chloroplast. Alright? So, two types of cells are present. One is palisade parenchyma, the other one is spongy parenchyma. Palisade parenchyma, where do we find them? Towards the adaxial epidermis. How are the cells shaped? They are rod-shaped, elongated. How are they arranged? They are arranged vertically parallel to one another. Spongy parenchyma, where are they placed? They are abaxially placed. So, what is their shape like? They are somewhat oval or rounded. How are they arranged? Very loosely arranged. There is a lot of spaces and air cavities present between them. Okay, so that's about the mesophyll or the cortex in dicot leaf or dorsiventral leaf. Now let's talk about its vascular bundle. In vascular bundle, here you will find both xylem and phloem in the same bundle. So the vascular bundles are conjoined. Okay. So the vascular bundles are conjoined. There's no cambium in leaves, so it is closed. Now, where in the leaf can we find these vascular bundles? Wherever veins are present, over there you can find vascular bundles. Generally, the size of the vascular bundles in dicot leaf will vary. Because in dicot leaves, if you look at the veins, there is a central midrib which is very thick. Its branches are relatively thinner. Its further branches are even thinner. So veins come in different sizes, right? So when you're taking a vertical section of the leaf, you can see that the biggest vascular bundle will be here in the center. It will be here in the center. On the sides, depending upon the size of the vascular size of the vein, you will have vascular bundles that vary in size. Alright, now in this vascular bundle, if this is the vascular bundle, see this is the xylem. Surrounding that you have the phloem. Surrounding that you can see there is a clear, distinct layer of cells that are made up of parenchyma. We call this the bundle sheath. Where else did we speak about bundle sheath? In monocot stem. There it was made up of sclerenchyma. Here it will be made up of parenchyma. Now, let's talk about. Yeah, that's pretty much it. Next, we'll have to talk about monocot leaf. We'll finish that also and then I'll ask you for doubts. Okay. So, a monocot leaf has another name. It's called isobilateral leaf. Iso means what? Iso means isogamy, you know, similar. Bi means two. Lateral means sides. So, isobilateral leaf, adaxial and abaxial epidermis will appear similar. There is no clear distinction between the upper and lower layers. So, it is called isobilateral leaf. Now, over here in the epidermis, you will find a single layer of parenchymatous cells as always, and stomata also will be present, cuticle also will be present. Stomata are present in nearly equal numbers on both adaxial as well as abaxial epidermis. You see stomata here in the upper epidermis as well as in the lower epidermis. So, stomata appear nearly equal in number in both surfaces. Okay. Here is an important point regarding monocot leaves. Generally in monocot leaves or leaves of grasses, what happens? Certain epidermal cells in the adaxial layer that are close to the vascular bundles. Okay, so the vascular bundle is here at the center. Certain epidermal cells that are close to the vascular bundles, they will get modified. They will appear slightly different from the other epidermal cells. How do they get modified? What is its role? They become modified by becoming bigger in size compared to the other epidermal cells, and they will become colorless and they will have vacuoles. Okay, so adaxial epidermal cells modify themselves into large and empty colorless cells. They have a special name. They are called bulliform cells. Now over here in a monocot leaf, you're finding stomata in the upper epidermis as well as the lower epidermis. If you've noticed, in monocots the leaves are not like this, it's usually like this, right? It'll be like this. If you've seen in grasses, it'll be like this. It does not face or expose itself too much to the sunlight. It'll be at an angle, right? So, whenever the conditions are not very favorable for the plant, there is a water stress or it's too dry, very less humid, not humid at all, and there's not sufficient water in the soil, transpiration will happen and water loss will continue to happen. To reduce the rate of transpiration, what happens is, these bulliform cells, they will lose the water that is present in it. They will let go of the water that is present in it. Once they lose water, they will become flaccid. Once they become flaccid, the leaves will roll. It will roll inward. Once the leaves roll inward, the surface area that is exposed to the external environment will reduce. Again, when it rains or when somebody decides to water that plant, water is available again. These bulliform cells will absorb water, it will become turgid and then it will become normal. So these bulliform cells exist to make sure that under stressful conditions for the plant, plants also have stress, right? Not just us. If water is not available to a plant, that is stress. If it's too hot, that is stress for the plant. If there is some pathogenic infection like a fungal disease or a bacterial disease, that is stressful for the plant. So like that, if there is water stress, until water is available again, these bulliform cells will make the leaves roll so that it's less exposed to the surrounding environment and less water loss will happen. Okay, so these are present only in the adaxial surface, not in the abaxial surface, along the veins, and they modify themselves into large empty cells that are colorless and they are called bulliform cells. Whenever they are turgid, the leaf surface will get exposed. Whenever they are flaccid, the leaves will curl or roll inwards to minimize the water loss. Coming to mesophyll, here there is no differentiation into palisade and spongy. Throughout it is parenchymatous, throughout they all contain chloroplasts. That's why in monocot leaves you do not see a color difference on the two sides mostly. They will be of the same color. Now talking about the vascular bundles, over here the vascular bundles will be nearly of the same size because in parallel venation you do not see any difference in the size of the veins. all of the veins will be of similar thickness, so the size of the vascular bundles also will be similar. Here, you can see xylem and phloem present together. We call it conjoined. Because there is no cambium, we call it closed. This is not mentioned in your textbook, but you may see this here, so I will tell you. If this is xylem and this is phloem, on top of the xylem and on top and below the vascular bundles, let me mark it in red. Over here, you can find sclerenchymatous tissue. These are made up of sclerenchyma and we call them bundle sheath extensions. Why do we call them bundle sheath extensions? Because they are extending till the epidermis on both sides. Okay, that's all about monocot leaf or isobilateral leaf. With that, we have completed this chapter. What do you think? It was easy, difficult? There's already enough in NCERT that is confusing, Gautam. Let's not introduce new terms and confuse the rest of us. Oh yeah, I just realized that I have not told you what stele is. Okay, so I'll tell you what stele is. Stele, not like S-T-E-E-L stele, S-T-E-L-E stele. This stele is all of the tissues that includes pericycle plus everything inside it. Pericycle, you'll have vascular bundles, you'll have pith if it's present, probably conjunctive tissue if it's a root, probably medullary rays if it's a stem. So pericycle and everything else that is present inside it is called stele. Okay? We are not learning about secondary growth. Correct, Kirtana. Telegram group name I think is called Yakeen Neet English 2025. Wait, let me check. Yes, Savita, I'll explain. I don't know. How do you guys join the Telegram group? Is there a link or something? Tell Sai Prasanna how to access. It's called Yakeen Neet English 2025. Yes, Savita, I'll explain. Bulliform cells are also epidermal cells. You find them in the monocots in the upper epidermis or adaxial epidermis. Basically, these cells will look different from the other cells because they are larger in size and they are empty and they are colorless. So these cells, what they do is when the plant is not getting sufficient amount of water, it will let go of water. Whatever water is there within that cell, it will lose it. When this cell becomes flaccid by losing water, the leaves will curl inward. Once these cells will absorb water and become turgid, the leaves will become normal again. Link has expired, okay. Alright, we'll discuss questions. We will discuss the poles also, but before that, let's differentiate between monocot leaf and dicot leaf. Dicot leaf is also called dorsiventral leaf. Monocot leaf is also called isobilateral leaf. Dicot leaf, adaxial epidermis does not have stomata. Abaxial epidermis has a lot of stomata. in monocot leaf, both epidermal layers will have nearly the same amount of stomata or the same number of stomata. Talking about the mesophyll, there is differentiation in dicots, there's palisade and spongy. In monocot, there is no such differentiation. Vascular bundles, both of them are conjoined and closed. In dicot, you have a parenchymatous bundle sheath. In monocot, you have sclerenchymatous bundle sheath extensions. In dicot, the vascular bundle size is nearly this, the size of the vascular bundles will vary, but in monocot leaf, the size of the vascular bundles is nearly the same except for the main midrib. Okay, now the size of the vascular bundles is uniform in dicot stem and monocot leaf. The size of vascular bundles vary in dicot leaf and monocot stem. Okay, keep that in mind. Today there is no DPP. I want to give you all some homework. Write down the differences between, make a list of the differences and send it to me. Difference between dicot root versus monocot root. Dicot stem versus monocot stem. Dicot root versus dicot stem. Monocot root you can send it to me on Telegram. Okay, on that group you can find me over there. You can send it. Somebody is asking something. Janani, why stomata is more in abaxial part? Because that is not exposed to the sun as much. It's present at the bottom. So transpiration rate will be low. I think I already mentioned that. Okay. So now let's start off with the questions. This is a question, brand new question from Neet 2024. Let's see how many of you will answer this correctly. We just learnt about it. Secondary growth is not important, Purnima. It's deleted from the syllabus. What are bulliform cells responsible for? Okay, the correct answer to this question is option C. 71% have answered correctly. Tawfiq leading the charts, top one. Then we have Vansh and Rakshita. Congratulations. Next question. This again is from 2024. Yeah. Tomorrow we will solve some questions related to this chapter and everything else that we've done so far, from living world, biological classification, plant kingdom morphology, Example based questions I will get for tomorrow's class. We will solve it. Okay. Don't keep typing it, Kirtana. You might lose access to comment. Don't type it. Which has thin outer walls and highly thickened inner walls? Which has that? It is the guard cells that has outer thin walls and highly thickened inner walls. So this image is not really very clear. A is showing other epidermal cells, B is showing your subsidiary cells, and C is showing the guard cells, D is pointing towards the stomatal aperture. So correct answer here is option C. Don't hurry, baby. 74% of you all have answered correctly. Shanti Priya from Om Shanti Om. And Golla Rangamma and Tiso, top 3. Endarch and exarch are the terms often used for describing the position of secondary xylem in the plant body. Exarch condition is the most common feature of the root system. Is the first statement correct? It's not the position of secondary xylem but primary xylem. Within primary xylem you have protoxylem and metaxylem. So it's not secondary xylem. First statement is incorrect. Second statement, exarch is the most common feature of root system both in dicots and monocots. That is true. So statement 1 is incorrect but statement 2 is true. Option B is the correct answer. Bad. 40% of you only have answered correctly. Marks, Vishwakarma. Hey, is this your real name or you've made it up, man? Okay, never mind. Tejaswini, top 3. Next question. So, Meghana, you'll get homework slide in the notes. You can check from that. Phloem in gymnosperms lacks. Phloem lacks what in gymnosperms? Both sieve tubes and companion cells, albuminous cells and sieve cells. Sieve tubes only, companion cells only. Lacks means it doesn't have. Gymnosperms don't have. They don't have sieve tube elements and companion cells. Instead of sieve tubes, they have sieve cells. Instead of companion cells, they have albuminous cells. So what they lack is sieve tubes and companion cells. Option A is the correct answer. 54% of you all have answered correctly. Nancy, Tawfiq and Sachin, top 3. Next question. Regeneration of damaged growing grass following grazing is largely due to lateral meristem, apical meristem, intercalary meristem, secondary meristem. Actually, this intercalary meristem is correctly pronounced as intercalary meristem, but my teacher taught it as intercalary, and that is how I will teach it. I will pass on the legacy. Okay, so the correct answer here is option C. Alicia, Nancy, Kavina, top 3. Next question. Meristematic tissue responsible for increase of girth in tree trunk. The correct answer is lateral meristem. Actually, phellogen is a type of lateral meristem, but never mind. This is a faulty question. Option B is the correct answer. C also can be correct. Taufik, Alicia, Ananya, top 3. Question is from my state. Correct. Correct, Taufik. It was from Karnataka NEET 2013. So you're saying my state is faulty because there was a faulty question. Prabhat, is it getting stuck for you? Is it getting stuck for anybody else? The chief water conducting elements of xylem in gymnosperms are? In xylem, in gymnosperms, which is the chief water conducting element? There are 2 water conducting elements, vessels and tracheids. Vessels or trachea and tracheids. I told you many times, several times, multiple times that wherever something is absent is given, you have to pay attention to it. We saw a question previously where they had asked about what gymnosperms lack. Now, the phloem of gymnosperms lack. Now you're talking about something gymnosperms lack. We learned that gymnosperms lack vessels. So obviously through vessels, transport will not happen. It will happen through tracheids itself, right? So again, it's based on that. Option D is the correct answer here. Oh my God, this is so disappointing. Maliha, Sashmita, Rakshita, top 3. Very lengthy question. Take a good 60 seconds and read it carefully and answer. Prabhat, try logging out and logging back in. In roots, xylem and phloem in a vascular bundle are arranged in an alternate manner along the different radii. Different radii in root, is it true? Yes, this is a correct statement. Conjoint closed vascular bundles do not possess cambium. Yes, that is also correct. In open vascular bundles, cambium is present between xylem and phloem. That is also correct. Vascular bundles of dicot stem possess endark protoxylem. Yes, that is also correct. Everything is correct or what? In monocots roots, usually there are more than 6 xylem bundles present. Huh. Where did I make a mistake? What did I not read correctly? It is in an alternate manner along different radii. That's why it's radial. Conjoined closed vascular bundles don't possess cambium. Where did I go wrong? Vascular bundles of dicot stem. Correct. Correct. Can you point out, can you tell me where we went wrong? Just give me 1 minute. I will quickly check. Usually there are more than 6. Correct. No, more than 6 vascular bundles will be present in monocot root. I can't figure out what the answer to this question is. So I don't know what to do. I'll check it and I'll let you know later. Okay, in tomorrow's class maybe. For now, let's see the next question. But if I mark any random thing, I think your poll will be affected. How do I undo this? I don't need this poll. Right. Okay. I'll randomly select A. Cool. Next question. Cells with active cell division capacity are called meristematic cells. So P can be matched with 2. Tissues having all cells similar in structure and function are called simple tissues. Tissues having different types of cells is complex, but they've not given complex. Instead, they've given vascular tissue. So that is correct. Dead cells with highly thickened walls and narrow lumen. Highly thickened walls and narrow lumen. What else is left? Sclerites, a type of sclerenchyma. Correct answer is P2, Q4, R1, S3, which is option B. Okay, not bad. 86% of you have answered correctly. Tawfiq, Nancy, Sakshi, top 3. Stomata and grass leaf. Actually, the question should be not stomata and grass leaf, it should be guard cells in grass leaf. Guard cells in grass leaf is of what shape? Grass is a monocot. Since grasses are monocots, you find dumbbell-shaped guard cells. Option A is the correct answer. 84% of you all have answered correctly. Alisha, Taufik, Rakshita, top 3. Rakshita is becoming a new competitor for the top spot, I think, from the last class, right? Last time we did a poll also. Rakshita was answering very well. Specialized epidermal cells surrounding the guard cells are called. Specialized epidermal cells surrounding the guard cells, what do you call them? What is the correct answer? Subsidiary cells, option D. 79% of y'all. Nancy, Vaishnavi, Sharmila, top 3. Vascular bundles in Vascular bundles in monocots are considered closed because correct, because there's no cambium. If there's no cambium, they're closed for secondary growth. Option D is the correct answer. 80% of you. Nancy is on top. Tough competition between Nancy and Taufik, and then we have Ananya in the top 3. Next question from Neet 2011. Ground tissue includes, what does it include? This should take you like 10 seconds to answer. Ground tissue includes all tissues except your epidermis and vascular bundles. So option B is the correct answer. Nancy, Taufik, Alisha, top 3. Alisha is also doing very well. Some vascular bundles are described as open because these are surrounded by pericycle but no endodermis, are capable of producing secondary xylem and phloem, possess conjunctive tissue between xylem and phloem, are not surrounded by pericycle. The reason is because they are capable of producing secondary xylem and secondary phloem. We've not learnt about secondary growth. This is a very old question. What basically happens in secondary growth is you have primary xylem on one side, primary phloem on the other side. In between that you have a strip of cambium. This cambium will undergo division and give rise to new xylem, new phloem that we call secondary xylem and secondary phloem. That happens because of activity of cambium itself. So the correct answer to this question is option B. Okay. 52% have answered. Nancy, Taufik, Raima, top 3. In barley stem, how are the vascular bundles arranged? Barley stem, stem of a monocot plant. Are they closed and scattered, opened in a ring, closed and radial, open and scattered? Barley stem, barley is a monocot plant. So in a monocot stem, we just discussed this today, in a monocot stem, how will the vascular bundles be? Is it open or closed? It's definitely not open. You can eliminate both of these options. It's closed. Is it radial? It's not radial. Obviously, it's conjoint closed. It can't be radial. It is closed and scattered in the cortex. So option A is the correct answer here. 55% of you have answered. Taufik, good job. Really, I'm proud of you. Maliha and Shivani, top 3. This is from 2020. ಏನಾ ಬರ್ತೀನಿ ಒಂದು 5 ನಿಮಿಷ. The transverse section of a plant shows the following anatomical features. TS, large number of scattered vascular bundles surrounded by bundle sheath, large conspicuous parenchymatous ground tissue, vascular bundles are conjoint and closed, phloem parenchyma is absent. Which part is that? It is a monocot stem. So option A is the correct answer. 82% of you will have answered correctly. Sakshi, Maliha, Nancy, top 3. So there are a few other questions you can do as homework. Try answering this. One of my old students is visiting me today from 2015 batch. So he used to live somewhere else in Karnataka and some other part of Karnataka. Today he's come to Bangalore, so he's visiting. So he's waiting for me outside. So I have to go. Okay. We have like 5 more minutes left. So you can do the rest of the questions. Try it and post your answers in the comments. Where do we find Casparian strips? Casparian strips are found in the endodermis of roots. So option D is the correct answer. 50% only. Nancy, Charan, Taufik, top 3. This is an interesting question from Neet 2014. Let's see if you can answer this. You are given a fairly old piece of dicot stem and dicot root. Dicot stem and root is given to you. How will you distinguish between the two? Based on what can you distinguish between a dicot stem and a dicot root? Oh, sure, Pratik, you're welcome. After your NEET exam, regardless of what the results are, you're welcome anytime. Hopefully, you will get to do whatever your dream is. I will make nice sambar and bhaji and everything for you when you come. we can find out from the position of protoxylem. If protoxylem is facing towards the pith, then it is stem. If protoxylem is facing towards the periphery, then it will be root, right? So based on the position of protoxylem, you can find out. Janani, Sai, Shri and Shanti Priya are in the top 3. There are a few more questions which you can do as homework. Very direct, not difficult questions, like 4 or 5 of them. Answer them without fail and post your answers in the comment section of this video. Okay? Yeah. So with that, we will finish today's class. Thank you. Bye-bye. Also come prepared for tomorrow's quiz. We are doing everything, all the chapters from living world to anatomy, all the specific scientist names, names of examples, everything you have to learn and come. Okay? Oh yeah, let's take a look at the leaderboard finally. Ajay is on top of the list. Then we have Rakshita, Yogita, Rangamma, Sai Prasanna, Ananya, Diya, Harshita, Shoritra, Janani. Okay. So thank you. See you all tomorrow, sharp at 12 o'clock. Bye-bye.